Hey, Bob, thank you for coming in. You're just a remarkable career. I mean, over 1,500 patents, founding or co-founding over 40 companies, one of nine institutional professors at MIT, which is the highest honor, over 450,000 citations, the most in history. I mean, what haven't you done? Just an amazing giant in biomedical innovation. So, again, thank you for coming in and doing this interview.
Well, it's my pleasure. It's an honor for me to do it. Thank you.
So, Bob, there's a remarkable career. Maybe what are one, two, or three inflection points? And it could have been very early when you were 5 or 6 years old to something happened in school, inside, outside the family. Could have been some early career moves or that really created this wonderful person you are today because you're very thoughtful. You're a servant leader, plus you're an amazing innovator. Where were those inflection points that inspired you to be who you are today?
Yeah, well, that's a very good question. I mean, I suppose obviously one of the keys is my parents, both the people they were, my mom was a super nice person, and my dad, I mean, he was very nice too, but he, would play math games with me, and they bought me these science sets, Gilbert chemistry set and Gilbert erector set, things like that. And I think that got me somewhat interested in science. I with the chemistry set, I even had made a little lab in our little basement. So that was certainly one. I think a second one that really was my postdoctoral work. I had done work in before that, like graduate degree and undergraduate, but I really wasn't clear what I wanted to do. And I think that what happened in my postdoc, I worked with a man named Judah Folkman. He was a surgeon, and I was the only, engineer or chemist in the entire hospital doing research. And I learned so much because I had such a different background from everybody else. And he also was a wonderful role model. He believed that anything was possible. He certainly was criticized a lot. People didn't think his ideas were right or other things like that, but he, hung in there. And I think him being a very good role model and him also, having big ideas and not never giving up, that was very, very important to me. And I suppose if I was going to pick one other, I when I was a graduate student and I was supposed to be doing my PhD, which research, which I did do, but I got very involved in creating this high school, alternative high school for low income working class kids in Cambridge, which Cambridge at that time, in the early 70s, had the highest dropout rate of any city in the country. And I got very involved in creating new math curricula and new chemistry curricula. And I think that it got me very interested in teaching and probably helped lead to me being a professor today. I still love working with students, and so maybe those three would be good inflection points.
And that's really fascinating. Yeah, within the family environment, you were encouraged to explore, to think in a creative fashion. You had mentors in your life that really said perseverance and kind of think out of the box and that narrative of grit. I guess it kind of the Angela Duckworth idea of grit. The perseverance, optimism. You don't give up. You just keep going. Those are all hallmarks of your career, and as a result, you've done just simply a really amazing work. Now, let's bring us up to the present. What are those areas that really excite you, and in the biomedical innovation area globally that you think are kind of the next areas that the world should look at?
Yeah, well, I break that up into a couple of categories. First, what's happening now and can be maybe next generations of them, and two, brand new things. And I think the things that are happening now that are exciting are, to me, are genetic therapies, which includes gene editing, it includes messenger RNA, it includes siRNA, which is short interfering RNAs. These are really, even though they're very young technologies, they've already made an enormous impact on the world. I mean, the Alnylam, which has been pioneering the development of siRNA. I was on their scientific advisory board from the beginning for the first 18 years. I mean, they have now five different drugs approved and treating many diseases. Moderna, of course, which I helped start, created a vaccine which, saved millions of lives according to various sources. And the CRISPR work gene editing work is early, but that's incredibly exciting, for treating different kinds of rare diseases and genetic defects. I think a second big area are cellular therapies. Cellular therapies include therapies like where you modify a cell, like a CAR T cell, to treat cancer or some other disease. It also includes an area that our lab helped do a lot of the early work on, which is regenerative medicine. Can you make new tissues and organs? Can you make organs and tissues even on a chip that might accelerate drug testing? And so those are two big areas. And permeating all of these things, of course, are things like artificial intelligence, which really isn't new. I mean, people sometimes think it is. It's actually gone on for the last 80 years. But it does accelerate discovery and things like that. And so that will certainly be a big help to so many of these things in terms of medicine and other areas too. I think in the future, the things I would maybe break that up into two categories too. One is work we're doing with the Gates Foundation, trying to create new medicines, better nutrition for the developing world. Even though a lot of people benefit from what's happened in the United States and other countries, still in the developing world, there's people die. I think something like just 20,000. There's, I think, a study out of Davis, 20,000 people die each year, mostly children, from not getting enough vitamin A in just Nigeria alone. And that's true for other places as well. And there's not enough iron and other things. So we've been working with the Gates Foundation to try to come up with much better ways to deliver micronutrients. We've also been working on ways to create vaccines that you might only need to inject once, to create pills that you could swallow and they might last for the entire course of treatment, like even a month or more. So that would be one set of things. I think another set of things are just things that are what I'll call curiosity driven, that it doesn't have an immediate obvious application, but it's something that, and it doesn't maybe have a name because it hasn't been discovered yet. CRISPR actually is a really good example of that, right? If you ask somebody 14 years ago what CRISPR was, they probably would think it had to do with cooking, but that's ended up having a tremendous impact. And I'm sure there'll be more things like that, but we just don't know what they are. But I always want to encourage people to follow their curiosity, to follow their dreams.
It's really interesting. I mean, you talk about CRISPR. I mean, Casgevy got approved by the FDA in 2023, and I believe there's something like 115 patients in the pipeline. You talk about AI in silico medicine has, I guess now their AI designed drug in phase 2A clinical trials this year. So it's isomorphic cloud spitting out at Google based on AlphaFold 3, but there's all of this AI embedded in it. Are you on the AI side? You can definitely accelerate everything that you talked about. Do you have any kind of a concern about where AI is going? Like Jeff Hinton has been out there this year. I interviewed him earlier this year. Quite a difference from, let's say, Jan LeCun, who believes that it's not quite there from an LLM standpoint. He believes we have to be hybrid models and things like that. Do you have any viewpoints? Or you just think it's just going to be great sort of opportunities, and as long as we address the safety, the ethical, regulatory risk and governance side?
Yeah, well, that's a great question. It's interesting. I don't know if you've interviewed Reid Hoffman. He's LinkedIn, and he just sent me this book, and I thought that he wrote with another person. When I first got it, I thought, great, I'll read it when I get a chance. Then I looked at the back cover, and the back cover actually has a picture of me in some kind of suit. Quotes from Shakespeare and from Tesla, actually about me. I thought, what is this? And then I opened up the book, and there's a foreword that I wrote about the book, apparently. Of course, I didn't do any of this. And then there's about 50 pictures of me in different things, even a poem I wrote about the book. And then I read the book, and which is a great book. And I think I think he made some wonderful points. I think he divided people up into like four different areas. People who are tremendously afraid of AI. I think he had doomers, gloomers, bloomers, and and so there was a spectrum, which is what you're saying, but I thought the point that he talked about, which was more of being a bloomer, if I get this right, was that this has happened throughout history, in his opinion. And what he used as examples is like, well, when cars first came, a lot of people thought this is terrible. And people even say today they cause pollution, they cause accidents. He said, well, yeah, but before that you had horses and there was manure all over the place and there was horse pollution. And then he went into GPS and he said people are going to be really worried about that. They'll know exactly where you are and they could kill you. But of course, that's ended up also help. So on and on, he pointed out that these things keep happening, and probably what needs to be done is to let them happen, but let them happen and grow with them and build in regulations that make sense, and ultimately the positive effects would far outweigh the negative effects. So anyhow, he obviously thought it a lot more about it than I did. But I felt what he said was in that book was incredibly thoughtful.
Yeah, I mean, it's really interesting. And in fact, if you went back, let's say 10 years and you went to the majority of researchers, they would have said what we have now in AI would have been AGI. They would have considered it AGI, right? And of course, that yardstick or that kind of benchmark keeps moving forward because these things just become more capable,
Yeah,
and we use it daily now, or like a lot of people just use it daily. Do you
yeah.
use things like the deep research kind of capabilities now, like from Google Gemini has deep research, OpenAI has this capability called deep research, where these agents will go off and think for 20 minutes, and they'll lay some foundational reporting and things like that.
Yeah, well, my students might. But just to take a step back, so we've actually used AI. I mean, again, like I said, it's not new, and we are, or you could call it machine learning, or however you want to define it. But even in the medical area, 26 years ago, we started a company, myself and Nick Gallicanos, called Transform Pharmaceuticals, and our idea was basically that up until that time, the pharmaceutical industry, one of the problems with drugs, a lot of drugs are not soluble. 50% of drugs aren't soluble. Another problem is that a lot are not in the right, what I call polymorph or crystal form. So at any rate, I'll just take the solubilization issue. So what we did is Dave Putnam and Hungmin Chen, they were two of my students. They started there, and they tested adding thousands of excipients, many in combination. So they got an enormous amount of data. And of course, data is really the key to all these things if you're talking about AI. So what we did is we were able to take drugs, figure out using machine learning we'd keep iterating. Like we test thousands and we learn what structures work the best and what combinations, and then we'd make predictions from that about the next generation and the next generation. And the company ended up being very, very successful. It got acquired by Johnson and Johnson at like a 25x return for the investors in just a couple of years, and led to about 3 new drugs. But so I've always felt like AI or machine learning can be very useful. In our own lab, we're working with Dan Anderson about using it to help design better lipid nanoparticles for delivery. But the things you're talking about, I mean, by the way, I should point out I'm not the most competent computer person. So I don't myself use some of those, but people in our lab, they use all kinds of things. And I can't even begin to know their names, or I mean, the students, of course, I know their names, but I don't know the names of the all the different things that are used. Certainly, lots of people use ChatGPT. even myself, I can put in, ask, pan a question, and it has an AI answer, and so I do things like that. But I'm not a very fancy person when it comes to that. But lots of my colleagues and students probably are.
There's another area that would sort of have synergies with the kind of work you do, and that's the availability of exascale AI computing in the cloud, or just exascale at a period from what they call the hyperscalers. So that could be like Microsoft Azure or within Google or Amazon. And exascale, being a billion operations per second, and it's kind of unimaginable. You have NVIDIA, they have their Spark workstation. It's a petaflop, at about what, under $4,000. That's like a supercomputer on their desktop. But you have the Argonne National Laboratories and the Aura supercomputer. They've been able to sift through 50 billion molecules in 20 minutes. And I was talking to Bronson and Monsieur. I interviewed him earlier this year at Oak Ridge, and with their Frontier, they have an insight program where people can just freely use their supercomputing capability, and they can integrate that with quantum and AI. Are you using any of that, or I guess maybe some of your students maybe try to get access?
Well, the students use all kinds of all kinds of things. They have different types of machine learning or AI approaches or neural network kinds of approaches. I don't actually even know the exact names or companies that make them, but we've had a number of students do theses on using these approaches at a high level.
Yeah, and I mean, it's interesting. Zetascale. I mean, it looks like Japan is going to build a physical, at least an AI physical Zetascale by maybe 2030, and it used to be thought that this was 2035. So, definitely it's going to help in any kind of biomedical innovation, things like that. How about quantum? Have you been following that at all? And do you think that's going to be kind of another major iteration that will impact your field?
Yeah, well, again, I'm not an expert on that. I think the answer to your question is probably someday. I guess the question is, what is that day? When I ask people, who are experts in AI, they tell me that that day is far away, I mean, 30, 40, 50 years. But, it seems to me people keep making advances there too. But I don't know that I really know enough to give a good answer or make a good prediction on that. But it's hard, obviously, the amount of information you'd get there would be, it would be a giant leap compared to even what we're able to do now, say base two. So I would think that someday I'm sure it will. I mean, I can't imagine that it won't, but I just don't know how fast it'll come.
Yeah, it's interesting. I mean, Cleveland Clinic has already worked with IBM, because IBM, I guess, is forecasting they'll have fault tolerance in quantum, which means it's usable like production quality by 2029. And they're already doing translational research with IBM on that. So it's really quite fascinating. And in terms of the work you're doing, what are the sort of the really big ideas? Because you always have these outlier big ideas that become mainstream. What are the ones that you think are really stretching, that you're really interested and curious about? Maybe some additional.
Yeah. Well, one of the ones that we're doing is out working with Li Wei Tsai at MIT, who's a brain scientist, and Alice Stanton, who's one of my postdocs. What we've been doing is creating personalized brains in vitro, like on a chip. So we've been able to actually do that. We'll have a paper coming out in a month or two. It's already accepted, but it's very exciting to me that we could take anybody's cells, convert them into what are called an iPS cell, and then convert those into any of the 6 brain cell types. And then actually Alice figured out a way to reorganize them in a way to actually, with the right kind of matrix, so they'll actually mimic a brain, have a blood-brain barrier, have the same kinds of structures. And so that, to me, could be a tremendous tool for drug discovery, for understanding new targets for diseases like Alzheimer's or ALS or other things. And of course, the beauty of it is that unlike humans, which you can't really do that many experiments with, or animals which don't necessarily have the same brains as we do, here we'll be able to do literally thousands of experiments. By the way, AI will help analyze those too. But so I think that coming up with these personalized brains and other tissues and organs, that's certainly very exciting. But the brain in particular is exciting because there's so many diseases that are, really just bad diseases, and if we could make a dent in those, that would be a wonderful thing.
That's really, really fascinating, especially because the brain is so complex. And if you can create these brain organoids and create a little bit of scaffolding, so you get this sort of more 3D matrix, then you get them simulating somewhat brain functioning. And as you indicated, then it can help with all sorts of, discoveries and things like that without impinging on people, right? And you can do this in the lab. So in vitro. So that's really quite amazing. Is there any other thing that you think is really, really interesting and then you want to surface here?
Well, we have another thing with gene editing that we did just, we'll have a paper coming out very soon on this with Phil Sharp and Vikas, who was one of our postdoc who really did all this. Is one of the exciting things about gene editing is that you can do what's called prime editing, which is a very powerful approach. But one of the issues with gene editing is it's not always error-free. In other words, you get what are called indels, and so you get some, you could get from that sometimes, possibly if you used it clinically, possibly safety issues. So because he actually come up with a way to essentially eliminate those, or at least reduce it by a tremendous amount by engineering some new kinds of prime editors. And that I also think will be a very big deal. That could make this field, it was already tremendously exciting. I hope even more so.
Do you think then that some of these could be applied to help with aging and things like that as well? Or do you see that coming up? Or
if we understand it well, I think what with aging is understanding it well enough. But the answer to that would be if we understand certain things enough, then sure, yes. But the issue on so many of these things is understanding the biology. If you understand the biology, I think you can move very quickly. If you don't, it takes a long time. I remember the board of directors of Glaxo came to visit Boston, and they said, would I give a talk to them? And I did. And the chairman said to me, he said, well, how come you could figure out, because he knew I'd helped start Moderna, a treatment or a vaccine for COVID in less than a year, and people have worked for 40 years and don't have a good treatment for Alzheimer's. My answer to that was just what I mentioned to you. I said, well, you knew exactly what you were going after in the case of COVID. You were going after the spike protein as a target, and making a vaccine that would, affect that. And with respect to Alzheimer's, we still don't really understand the biology well enough, and that I also think is true for aging. I mean, we understand some things at a cellular level, but there's so much that we don't understand. Now, there are companies working at that too, and there's certainly lots of scientists working at it too. But I don't think but as people learn more, then I think we'll be able to do more in terms of whether it's gene editing or mRNA or other kinds of approaches.
Well, you definitely are leading the world in so many respects. You've got over 400 companies licensing or sub-licensing your patents, and that's just going to continue to grow just from all of the, the marvelous work that you do. And I believe you have the largest lab at MIT as well in this area. So just thank you for coming in. And I just have one final question in the remaining minutes. What recommendations would you like to make to the audience, and or questions you want to leave the audience?
Well, I think, I think recommendations, to me, what I think is great about science is really the good it can do. First, I think it's great to learn things and to create new knowledge and to do curiosity-driven research. And I think anybody that wants to get into science, I mean, that's great. But I also think that once you learn some of these things, there's a tremendous opportunity to do good, to create better cures and better treatments. I also think that the more informed society is about science, that's a very, very useful thing. I mean, one of the things that's going on in the United States now is there's, decisions that are being made not necessarily on the basis of science, but on the basis of politics. And I was chair of the FDA's science board. We made a point of the whole, everything should be, if you're talking about people's health or things like that, you want things to be science-based. So I just feel that the more we can bring in, the more people that enter politics that have a good science background, the more people that in industry and every field, I think understanding science and doing things that are science-based, I think is great. And I think it's a wonderful career. I think whether you're a teacher or a professor, working in companies, working in hospitals, I just think there's so many things that you can do with science.
And then, because the audience is so big, I mean, I have CEOs in my audience and students and also scientists and engineers and investors. Is there a question or one or two questions you want to leave them?
Well, I mean, I guess people could think about what's the next big thing, which was a question that you asked in different ways. And I think that's a question we should all ask. And there probably is not a single answer to that, but that's okay. I think that that's actually but what things, if you had to think about, and I sometimes think about this for my own work, if because you only live so long, what can you do with the time, with your own skill set and the time you're here, that can have the biggest positive impact on the world? I think that's a good question for everybody to ask themselves, and then to try to do the best they can to do that.
Well, Bob, you definitely are an exemplar of all of these things. And thank you for coming in and sharing so many of your insights and innovations with our audience. I encourage the audience to follow your work. It's just amazing, outstanding, and it continues to, resonate throughout the world. So thank you again for coming in.
Stephen, thank you so much. It's just a pleasure and an honor to speak to you. Thank you so much.
Thank you for listening to the Brand Called You videocast and podcast, a platform that brings you knowledge, experience, and wisdom of hundreds of successful individuals from around the world. Do visit our website www.tbcy.in to watch and listen to the stories of many more individuals. You can also follow us on YouTube, Facebook, Instagram, and Twitter. Just search for the Brand Called You.